afternoon. Thank you for joining us today, and welcome. Today we are honored to have our own very Dr. Anthony Lyons. Before I pass it on to him, I'll just quickly go through his bio. Dr. Anthony Lyons is currently a research professor in the Center for Coastal and Ocean Mapping and is the Associate Director for Research in the Center for Acoustic Research and Education. He received the BS degree in Physics, summa cum laude from Elderson State University, Acoustics, in 1988. After receiving his PhD in oceanography from Texas A&M University in 1995, Dr. Lyons worked in the fine-scale acoustics and oceanography group at the Circlant Undersea Research Center, La Spezia, Italy, until 2000, where he was involved in a variety of projects in environmental acoustics and oceanography. Before taking his position at the University of New Hampshire, Dr. Lyons was a senior scientist at the Applied Research Laboratory at Penn State and a professor of acoustics in Penn State's graduate program in acoustics. That's from 2000 to 2014. While on sabbatical, he worked in the electrical and computer engineering department at the University of Canterbury, Christchurch, New Zealand in 2008. Dr. Lyons also worked for the US Office of Naval Research Global as an associate director in 2008 and 2014, and was a visiting scientist at the Center for Maritime Research and Experimentation, a specialty in 2023. His research projects have included studies of shallow water acoustic propagation, acoustic interaction with the seafloor, characterization of seafloor sediments, and high-resolution oceanographic measurements. Dr. Lyons was awarded with the recommendation of the Acoustical Society of America, the Institute of Acoustics, a UK AB Wood Medal in 2003. He is a fellow of the Acoustical Society of America 2007 and a member of the IEEE Oceanic Engineering Society. Please join me in welcoming Dr. Anthony Lyons.
Thank you, Elias. I was looking back, the last time I gave a talk in this Friday seminar, it's 2016. So none of the students really know what I do. They may see me walk in the halls, but they probably don't know what I do. A lot of the faculty probably don't know what I do. So I'm going to talk about a couple of the things, thing one and thing two, that I've been doing recently. And both of these relate in some way to synthetic aperture sonar, which is a thing that I've been studying for a while. So just a quick overview, and I'll give you a couple more slides later. Synthetic aperture sonar is a technique, it's a sonar system, but it's a technique that yields really detailed, high-resolution images of the seafloor. They're almost photographic quality. And I'll explain how that works in one slide in a few slides. Okay, so I'll give you a quick overview of SAS, and then I'll head off into thing one and thing two. Okay. In my career, which has been long, I do a very good job of collecting people to help me with things that I'm interested in. So these are all the people that have been kind of involved in the stuff I'm going to show today. Number one is Jenna Hare, who's sitting right here, and she's been involved in a lot of the quantitative results that I'm going to show today. Nick Lamanna was a student here at CCOM. He graduated in 2023. He's now with Ursa Space Systems, which is a synthetic aperture radar company. Roy Hansen and Torsten Seibo are with the Norwegian Defense Research Establishment, or FFI. They've contributed data, and also we've analyzed and written some papers together with the data that I'm going to show you. Shannon Steele was another CCOM student here. She's now with Kraken Robotics. She graduated in 2019, and I just worked with her about 3 weeks ago on some of the stuff I'm going to show you at the very end of the talk. Dan Brown was another former student of mine. All these, Nick and Dan and Shannon were students of mine. Dan is at the Applied Research Lab, and he provided some of the signal processing expertise that we use. Jan Pela is at the NATO Center for Maritime Research and Experimentation. That's where I used to work. It was named something different 30 years ago. And I was just working with him, and I'll show you some of the data collected with him. And Tim Marston, I'm going to show you some results that he did. They're all his, not mine. We're hoping to maybe collaborate with him in the future based on what he's seen. So I'm going to show some of that. If he's out in the world watching this, thank you, Tim. So just quickly back to this slide. So synthetic aperture sonar is, again, as I said, a technique for making really high photographic light pictures of the seafloor, but they're really just pictures. They're uncalibrated systems, so it's qualitative. So what I'm going to do today is show, I think I know of probably only 2 studies that have gotten quantitative data out of synthetic aperture sonar systems of the water column and processes in the water column, and that's these 2. So it's not a typical thing you do with SAS or SAS data. Whoops. All right. So I'm going to give you 2 slides. One is side scan. So this is the old technology for making images of the seafloor. And it's a lot of you might know this, but it works by making sort of raster images of the seafloor by pinging a nice narrow beam, moving a little bit, either towed or on a vehicle, pinging, collecting that scatter data, and you form an image that looks something like this. If you want a high resolution image, it's only an angle. So you've got a single physical array, you get resolution in angle. So as you go out in range, you get a range dependent resolution so that when you're at the far ranges, it's pretty crappy. You may have a 1 degree beam and you may have 5 centimeter resolution in here. You're going to have 75 centimeter resolution out here. So that's a drawback of side scan sonar systems. You can increase the resolution, but you have to increase the frequency. If you go up to 500 kilohertz or 700 kilohertz, you're automatically going to reduce the ranges that you can see because of absorption. So these are some of the drawbacks. This is technology that's older than me, not a lot, but a little bit. Okay, in the last 10 to 20 years, a new technology kind of came out or was developed, and the theory goes back further, but this was kind of proven about 20 years ago, and then 10 to 15 years ago, they started being commercially available, these types of systems. So relatively new. So this is what's state of the art in imaging the seafloor. Complicated, expensive systems, but this is kind of the quality you get. So this is the same image that we saw here. They've just made it sharper by using a bigger array. Now, how did they get that bigger array? Well, instead of just having a single transmit receive on a single array where they get resolution and angle, they don't process the data. So synthetic aperture sonar doesn't process the data until they've collected a number of pings, 10, 20, 30, 40. So you can form an array that's tens of times larger than the physical array. There is a trick. The reason this took so long to sort of turn into a practical application is because you have to straighten your array to sub-wavelength. So imagine you're at 100 or 300 kilohertz. It has to be straight to sub-millimeter resolution. But there are data-driven methods to do that. And I won't go into that. If you're curious, you talk to me or Tom Blanford, and we can tell you what that is. But it allowed, you can use lower frequencies, so you can get further out in range. You can actually get constant resolution with range. And how do you do that? Well, at far ranges, you can use a long synthetic array. At short ranges, you can use a shorter synthetic array. So you get the same resolution here as here. So this might be a half degree beam here. Out here, it's a tenth or a hundredth of a degree. So you can play that trick. So you can get square pixels or regular pixels, but they don't degrade with range. This is another advantage of SAS imagery. Okay, it's obligatory when talking about SAS to show pretty pictures of shipwrecks or plane wrecks on the seafloor. And so here are my obligatory images. So I'm actually showing data today from high SAS systems, a lot of data. And I'm going to show some data from the Kraken system. So these are 2 commercially available SAS systems that you can go out and buy if you have a couple $1 million, $4 million, but they're available. But you can see here the resolution. You can see all the framework of the wings on this guy. You can see the detail of the wreckage here. I was on this cruise. This was on the Oceanographic Explorer. It was a tech demo cruise for this. This was a towed SAS system.
Looks like the cruise didn't go well.
Okay, just one more example because I like this one. We found this one with a side scan sonar back when I was in Italy. They went back a few years later with this mussel system. This is actually the grandfather of the Kraken system because it's the same sonar designer who left and went to Kraken from CMRE. This is a Roman shipwreck here. The only thing that's left are these clay pots. These are called dolia. They're taller than me, much wider than me. So these ships used to go around the Mediterranean. This was called the supertanker of the Roman times. And look, you can look at the images and the photographs, and these are really close to photographic quality. So that's another advantage of these. So these SAS systems are used for making sharp images of the seafloor, mostly not really to do a lot of archaeology, but that's a good thing we can do. But often the Navy uses them to find objects on the seafloor. So you want the highest resolution you can get. Okay, let's go into thing one. So this is the first sort of topic I'm going to talk about, where we get quantitative measurements using a side scan sonar. And this is some fun stuff. So I don't have a presenter mode, so I'm guessing what slide comes next. So I'm going to take you back in time. It's 2012. It's Christmas time. Well, this was earlier. This is October 2012. My friend Roy, who's with the Norwegian Defense Research Establishment, takes this system out. This is a Hugin AUV with the high SAS 100 kilohertz system. It has about 3.5 centimeter by 3.5 centimeter resolution, all the way out to 160, 170 meters. It's phenomenal. The experiment took place off of Elba Island, and you can tell that's Elba. Why? Because who's exiled there? Yeah, Napoleon, he's there, you can see him. Elba is off the Tuscan coast, and you can tell it's off the Tuscan coast because Pisa is right here, and you can tell that's Pisa. Right? There's my brother, he's kind of tilted as well. Okay, so he didn't experiment in Elba Island. And he sees this. He sends this to me. I'm on Christmas break. He sends this to me and he goes, "What do you think about this image?" And I said, "I don't know. Looks like a sandbar." So that thing is, I don't know, 60 meters long and maybe it's like 3 meters wide. And I said, "Oh, it's a sandbar. It's the depth is, I don't know, 40 meters, something like that." Made sense to me. So he sends me another image the next day. And it looks like that. This was taken the next day. And this is flat calm. In October, it's usually really nice in Italy, flat calm, sunny, and it's gone. So I said, well, I don't know. Well, actually, I had an idea. So another story. When I was in Italy in the late 90s, digital cameras, the point and clicks that were digital, had just come out. And so I did a lot of work off Elba Island. We went there all the time. So one time when I was there, I had my brand new, like 500, it was tiny resolution thing, or not a very good resolution camera, but I took it out on an experiment when I was at Elba, and I found this guy. So, I mean, right? So I think that's what it was, and I sent it to him, and he didn't buy it. So he sent me this image. He built a bigger image, and you can see that guy is here, and there's no serpent here, it's not a tail, but there are other features, and they come in sort of pairs maybe, and the color is bathymetry. This is a long track, the way the vehicle's moving. This is range away from the transducer. These sandbar-like things kind of line up with the bathymetry. So I'm thinking, okay, that makes sense, sandbars, except they disappear after a day. So it wasn't until maybe 2 weeks later, he sends me, so what's going on? He sends me this data. So he sends me CTD data. So I look at a temperature profile, and it's very stratified, very stratified. It goes from about 22 degrees Celsius to about 15 or 14. And so if you've all had physical oceanography, or when you take physical oceanography, you know that on these very stratified water column, you can get waves along that boundary, along that water mass boundary between the warm, light water up top and the cold, heavy water at the bottom. So I'm thinking maybe this is what's causing it. So just a little background, internal, this is called solitary waves, but internal waves develop when there is usually a tidal effect over bathymetry, so you get a hydraulic jump. It makes a wave, and the wave propagates. I didn't know until this time that I mean, it's obvious now, but they actually can interact with the slope. These guys are sitting, I don't know, 10, 20, 30, 40 meters under the water. They propagate slowly, interact with the slope, they break, they mix, and then they produce these little bolus things. And here's a little movie, a GIF from this website, and you can see exactly what I'm talking about. Big internal wave, they're big, hits the slope, steepens, hits the slope, and then these boluses go crawling up the slope. And they're in sets of 1, 2, 3, 4, 5. And this is very typical. So after a little thought, these guys have enough curvature to form a lens. So just like light through a drop of water, where you have a bright spot, focus spot, and then a shadow, because all that energy has been moved into the spot, you get the same thing behind this bolus. And they have a shape that's sort of parabolic. Energy, well, let me just go to the next one. So the sound hits the top of that bolus. Because that sound speed, that cold water has a lower sound speed, it refracts down. So you get all these rays bending down. And because of the curvature of that, they all focus. It is an acoustic lens. It focuses at a spot. So if you think back to those images, we saw those bright lines. These boluses are big corrugations as the waves break on the internal beach under the water, and you have this focusing behind the entire wave. You were not looking at this, you were looking at where it actually hits the seafloor. So we do not see these directly, we see the bright focus region. Okay, congratulations, you've all been subscribed to Internal Wave Bolus Facts. So I'm going to repeat what I've already said. This image is from a paper by Bourgault from 2007. He took an acoustic system, an echo sounder, and went over this high frequency, I forget, 200, 300 kilohertz. He can actually sense the turbulence at the top of this bolus. And he sees the big shoaling wave, steepens, turns into a bolus. The bolus propagates upslope, getting smaller as it goes up because it's mixing with the surrounding water. So that's pretty cool. Why do we care? If there are several reasons. If I'm the Navy, I care because these bright spots and shadows can mess up a lot of the detection systems that are set up. If I'm an oceanographer, it's also important. So it plays an important role in mixing of these water masses and energy dissipation. They can trap and transport biology, larvae. They can suspend sediments and move them upslope. And I would guess, and I'll show you maybe an example later, these moving this nutrient-rich cold water with larvae and sediment can be vital for maintaining coastal ecosystems. And they've been observed all over the place. And I'll show you a lot of examples coming up. So this is Roy's data, again from Elba. It's a different location than before. Just to orient you, this is a pipeline. I've worked here as well. I'm not going to tell you what comes out of this pipeline, so don't worry about it. But these are images, SAS images taken from at the same location every sort of hour-ish. So 5 times over about 5 hours. And the seafloor is just alive. It's just moving. If you did not know, if you did not go back, you would not know this wasn't a seafloor feature. I mean, it wasn't, sorry. You would think it was a seafloor feature, not a water column feature. So this is interesting. These features can be large. So this is a SAS system, but it's operated in side scan mode. So you don't have to do the synthetic aperture part. You can just treat it as use the physical array to make a side scan image, and you can look at larger areas. And that's what Roy did here. So you can see they're kind of corrugated, they're long and linear. In this instance, they're not always, and I'll show you examples later. But they can be big. So it's about 450 meters for these guys. We've seen them on other systems. So this is the mussel system, which I showed you that Roman wreck. These are just 4 examples. And once you know what they look like, the flavor, you can look at images, you go, oh yeah, that's not a seafloor feature, especially when you see them cutting across ripples. So that's kind of obvious. This is a Kraken system, and they've found these all over. This is in the Arctic. This was part of that Okeanos tech demo cruise I was on where we found the shipwreck. This is in the Baltic, these guys here. So in these low angle systems, anytime you see sort of these internal wave effects, you're going to get your imagery kind of messed up. So it's kind of important for us to understand, you know, when these are going to happen, why they're happening, and that's what we've been doing. Okay, 3 weeks ago, I was with Jan and Shannon and Roy and Tim at Elba, and they were testing a new Kraken system that CMRE bought. And one of the first things they saw was these internal wave focusing effects. And it was just all over. So I'll come back to this in a second. This is pretty fun. This is a 350 meter by 170 meter, or actually about a 130 meter image. This is seagrass. This is sand. These are drag marks from anchors. That's one day. This is a couple days later. So you can see it's kind of dramatic. So back to sort of the thesis that this cold water is kind of washing over this ecosystem and bringing nutrients to this ecosystem. This is a good thing.
Tony, what's the water depth of that?
That water, all these are, I don't know, I don't remember exactly, but these are all sort of 30 to 50, 60 meters. All right, so these didn't turn out very well, but these boluses, this refraction also messes up interferometric bathymetry. So you can, in addition to imagery, you can use 2 arrays and look at the phase differences between the returns from the images and get bathymetry. So this is a combined map of the intensity, and it's easier to see here, intensity and bathymetry. Now, the seafloor doesn't look like this. It doesn't have these bumps. Those are fake. Those are artifacts of the refraction causing mistakes in the bathymetry. The interesting thing is, again, if you did a single pass, these are bright on the right side of the fake bathymetry and shadowed on the right side, the correct side of the fake bathymetry. So again, if you went out with an interferometric SAS system, there's no way you could tell this wasn't a real seafloor feature. So you have to do some other things. You have to make multiple passes, or there's some other tricks you can do, which I'll talk about. If you're interested in change detection, this is just the difference between those 2 images in that seagrass area where there was internal waves and when there isn't. And you can see there's a big intensity difference. I've chopped it, but it's more than 10 dB. It's a huge difference. If you're trying to find objects by looking at before or after pictures, it's going to be hard to do that in this sort of environment. This is the bathymetry difference. So this is the bathymetry before and after those internal waves were present, and it causes bathymetry errors in about 30 meters of water of about 1.5 meters. This is big, and these are not real. So that's going to mess you up. Okay, this is a little movie playing. So this is data provided by Jim Prater. He's at the Naval Surface Warfare Center in Panama City. They have a different system, the SM system, and he caught one of these off of San Diego. So if you watch, there's one, there it is, and there it kind of washes out. So the typical life is they're big, they get smaller, 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 and they kind of wash out, just like a surface gravity wave. This is speed. As it forms here, as it comes in, it slows down before it kind of washes away. And so our measurements are about right here, and you can see we see 9 centimeters per second, 7, 3. So when it's coming toward the end of its life, it's moving sort of lava lamp slow. Imagine 15 seconds to go across a distance about like this. Okay, so that's all pretty, and those are all kind of pretty pictures, but can we say other quantitative things about these boluses? Well, it turns out we can, and it's because of how the SAS data is collected, presented. So if you can see these little stars, there's a technique called parallax, which they use in astronomy to determine the distances of nearby stars. If you can make an image of the sky from different angles, you will see that that star shifts with respect to the background. And you can see a shift. And that tells you that distance. So we can hopefully we can do the same thing with the internal wave caught on the SAS systems. So it turns out, the lucky thing is that we can actually form multiple looks from a single SAS image. We can do that because this is a complex image. It actually has the phases included, and so we can go back and forth in between wave number space and image space and chop up the wave number spectrum and form different looks. So what you're seeing here, this little movie, is not these guys moving. It's us looking at them from different angles. So if we look from one direction, that bright point looks like it's here. If we move over to another direction, that bright point looks like it's here. So this is a disparity. This technique of looking at different angles is looking at parallax technique. This movie is made of about 20 different looks, so we're just marching an angle, and we're seeing that image slide back and forth as we look through that acoustic lens. Okay, just a quick recipe. How do we form the different looks? We have single complex images. Before you transform into the wave number domain, you can do a little massaging to fix the spectrum. Then you can just chop up your spectrum into a long track bands, reform images. They'll be lower resolution because you have less support here in wave number. And each of those new images corresponds to a different look angle, just like the movie that we just saw. So when I started this stuff, so this is Jenna's. She made this flowchart, and I loved it so much handwritten that I left it. When I started, I did a bad job on this whole slide. So I could get to this part, and I could get to the next part, but I got a pretty bad result. So Jenna, there's a lot of kind of image processing that goes on to get to the disparity part, where we measure the separation or the shift in the images from different looks. And you can talk to Jenna if you're really interested, but this is the flow chart of what has to be done. It finally worked when she got hold of this. So this is a visual of what's happening. So we take a full band image, chop it up into different bands, doing our little wave number space chopping up, and we get, in this case, maybe we had 16 bands, and I'm looking at band 15 and band 2, and it's not super clear because you lose resolution, but if you notice the shadow here is pointed this direction, so our eyeball is over here. The shadow here is pointed this direction, so our eyeball is over here. We're just looking at it from two different angles. Easier to see the shift down here. You see this feature? It's moved about 10 meters. That little kink is right there. That little kink is moved over here. So just because we've looked from two different directions, we've shifted where that bright focal region appears in the image. So Jenna will threshold this, turn it into a binary image, and we can just detect these little blobs. She calls them blobs, and I like that. And if you take the blobs here and the blobs here and just show them in red and green, that's here. So you can see they've shifted. It's an easier way to see the shift. Measure the shift, and you can get what's called disparity. That's actually the measure of the shift in meters, and you can see here it's about 10 meters for all these guys, as we saw up here. Okay, so what can we do with that? So once we know that shift, that actually tells us something. If we know the angles and we know the shift, that tells us something about the size of this guy. The bigger this guy is, the farther these are apart. And so we can use geometry here and here, this is my bolus, and it's refraction, to say how tall it is, assuming that most of the refraction happens at the top of the bolus. What we do have to know is this guy, the effective refractive angle, and we get that sort of empirically by running ray trace models. All right, here's the final result. You see here, this is my blobs, this is my image with the pipeline. Jenna has highlighted the different colors, which are going to, each of these is going to give a different height, and she's estimated the heights. This is the bathymetry. So if you take all these blobs and you look at the heights that we've estimated using the technique in the previous slide, you get this. So these guys are big. So they're up from about 13 meters in this image down to about 5 meters tall. And they're about the same, they're about twice the width, maybe a little more, because they're parabolic. So maybe about 10 meters wide, or about 30 meters wide, about 13 meters tall. So these are big. They do as we saw in the Borgo paper, they do decrease in size as they go upslope. So let's take a slice along that intensity image. And so you can see the red is the intensity. You can see we've got a spike for this first one, a spike for the second one, a spike for this third one, and so on. So if I take these sizes and this is the actual position of the bolus, not the bright spot, but if I take this position and this height and run a ray trace model, I can predict what I think I will get for the intensity, and we match that pretty well. So this is just to check that we're doing things correctly. So just to show you, we have several data sets. These are 5 of them. These are the images and the blobs that Jenna has found with her algorithm, color-coded again as before, and these are the heights and with respect to position that we found. And you can see, 13-ish, 13-ish. We already saw that. This one was down here around 1 or 2. This is over the seagrass areas, and they're up to 20, 15 meters. So these are big. Keep going fast. Okay. So the experiment that I was at just 3 weeks ago with Tim and Shannon and Jan and Roy, this is data. So this is fresh data. Jenna just gave me this yesterday. So these are the technique where she's identified these specific boluses, and then I'll show you the heights on the next slide. You can see there are a lot of them. So these are 5 different data sets that are contiguous, so she's connected these. There are a few things, that the algorithm missed, but a little tuning, maybe we can pick up more of those, but these are the larger features here. So these are the heights versus range to that acoustic lens or bolus, and you can see just what we kind of expect. Looking up slope, they're large when they first form, and then they become smaller and smaller and smaller, and eventually kind of go away, just like in the movie. A more interesting thing is this. This is volume. So imagine I'm just collecting. I know the shape of these. I know how long they are because we've calculated that. I can say what the volume is as a function of range for all these boluses. So I'm just collecting volumes as I go. You can see here, sort of further out in the deeper water, they're just starting to form. Here's when they are maximally big. They're the steepest they're going to get. And look at the volumes. The volume just in that area of the images is, each of these data points is about 1,000 cubic meters. So a better way to do it is to do the integration in this region where their peak, their biggest, it's about 30,000 cubic meters of water moving up slow, just in the image of about 20,000 square meters. They do then start mixing with the surrounding water, and you see that they kind of, the volume of the cold water decays, and then you'll see that it kind of swashes out at the end, just like the movie. All of this took about 5 to 10 minutes. So imagine this sort of lava lamp stuff coming up the coast, giving some nice fresh nutrients to the ecosystem. And this happens over and over every day when there's a nice stratification. All right, thing 2. I have time to do thing 2. Okay, this is a separate thing, thing 2, but it also took place actually 2 years ago. I was a visiting scientist at CMRE back in 2023, and they had just collected data off of Biotola, and I've worked off of Biotola many times as well. This is an image of Biotola Bay. This is sand. This is seagrass, where it's dark. So these are prairies of seagrass. I knew this was Elba because why? Yeah, Napoleon. And I knew it was off the Tuscan coast, right? Okay. So the equipment, this was a house-built synthetic aperture sonar. So they wanted to go really low in frequency, and they didn't want to spend the time building one themselves. So they went and got an off-the-shelf sub-bottom profiler. And we have some of these sitting in the high bay. These are 2 to 16 kilohertz, nice bandwidth. You want lower frequencies. They're thinking they might see into this grass and see targets because they're really paid by the Navy. You might see into sediments better with the lower frequencies. You might be able to image into targets. So there's a reason they want to go to this frequency. And they were just testing it. So they built their own using off-the-shelf components. And they had a larger receiver array, but they gave me this. And it was calibrated. It was partially calibrated by them and partially calibrated by me. You're going to notice the quotes. Don't ask me or ask me later. Calibrate. Trust me. It was oriented at an angle to the seafloor because that's how we do side-looking imaging systems. And this was also with Jan at CMRE. Now I'm going to fess up. This was done in a side scan mode. So I was not doing the actual beamforming at the end to get high resolution images. I was using it in side scan mode because I want to look at larger volumes. But it's still a SAS system, and I'm using the data from the system. So I can do this with any SAS system. Okay, so what did they see? Jan came to me, I'd been there 2 months, and in the third month he came just in the beginning of December, he shows me this, and I said, what is that? I love these when they, Roy or Jan come to me and they just show me stuff. And I said, what is that? And I'm like, I don't know what that is. This is in the water column. So I chopped off the seafloor data. You'll see stronger scattering down here. This is just in the water column. So we're pinging, just like a side scan. And this is range as we move. And there are times when we see in the water plumes of strong scatter. So what are those? Is it fish? Zooplankton? I don't know. But it didn't take long, again, when he told me where he collected the data. He collected it over seagrass. This is not from his study site, but this is eelgrass, but it's very similar to the seagrasses that he was collecting data over. And you see what's in the water column? You see what's on the leaves? These are bubbles. So there are plumes of bubbles causing scattering in the water column. And the scale here is 1 centimeter, so these guys are hundreds of microns. So maybe a tenth of a millimeter, maybe partial, fractions of a millimeter. So you can see in the water column, they're probably a little smaller than these guys on the leaves. But lots of bubbles. Bubbles scatter extremely well. If they're anywhere in your system, that's going to dominate. So that's what we were seeing. We're seeing bubbles in the water. Okay, congratulations. You're also signed up to Posidonia oceanica facts. Posidonia oceanica is a seagrass that only grows in the Mediterranean, but it grows everywhere along all the coast. And this green here is from this paper. This is all the places they've noted it. It actually grows everywhere, but this is where it's been noted. Interesting facts. It is the world's largest clonal plant. Don't believe those aspen guys in Colorado. It's actually this guy, up to 15 kilometers wide, a single plant. So it's a grass. It can spread by sending out rhizomes horizontally, so they're single organisms. Some of these colonies are so huge and so old, they can be up to 200,000 years old. 15 kilometers wide, 200,000 years old. It makes them very sensitive to environmental change, though, as I'll get to in a second. They produce, these guys in the med produce twice as much oxygen as the Amazon rainforest. Lucky Italy, yeah. They can store carbon 15 times more efficiently than a tropical rainforest. The economic value for the environmental services they provide is 10 times higher than tropical forest. Economic services is everything. It's habitat, whatever. Bad news. These red areas are, again, areas they've noted, but it's probably regressing everywhere, where they've noted regression of these seagrasses. These, just like the Amazon rainforest, as we lose this, we're losing this sort of the lungs of the planet when we do that. So the estimated regression over the last 50 years is about 34%. That's huge. And that's due to pollution, turbidity, rising water temperature, the anchor damage that you saw in the images. So this is not a good thing. So people are interested, and they want to measure these environments. They want to make some quantitative measurements of sort of the photosynthetic activity to see how much biomass might be produced. So it's very difficult to do that. Usually they send divers down. They have sensors, dissolved oxygen sensors. But maybe we can do better. So again, this image from this paper by Long if you read fast, you can read the whole thing. If you read slowly, read the red. If you can't read, just look at the picture. The point of this is the studies that have tried to quantify photosynthetic activity usually use, or always use, dissolved oxygen sensors. They're missing half the problem if you've got a lot of free gas on the leaves and floating in the water. So this paper is saying, maybe we want to study that. They did it with big traps, big cones that just collected the bubbles. If that is a small area, point measurement, maybe we can do better. Those plumes are one key. If we can quantify that, maybe we can do better. So what are you seeing here? So what I showed before was an image of the entire 2 to 16 kilohertz band. What I'm doing here is band passing it to a 2 kilohertz bandwidth. So I'm just looking at 7 to 9 kilohertz here, center frequency of 8 kilohertz, a different plume. So again, this is range away from the sonar in the water column as we move along 200 meters of seafloor, or above 200 meters of seafloor. I'm going to talk about some data here in this red box in a second. So you're going to see no data, no data, no data. You hit the plume, you're going to see data. So that's shown here. This is 8 kilohertz. And if you look at this gold symbol, that's this guy. That's the strongest. The way to read this is range. Again, this same range, no data, no data, no data. You hit the plume, and I get volume scattering strength. So this is a real measurement. This is volume scattering strength with a calibrated system. We go up for the 8 kilohertz up to minus 34 dB, which is strong scattering in the water column. If you've ever collected data in the water column, this is honking. This is huge. So 8 kilohertz is the strongest. If you go down to sort of these guys, that's 6, 10, 12. They're a little lower. If you go to 14 and 4 kilohertz way on the outsides of the bands, you see it's even lower. So can we do something with that? And the answer is yes. And why? Because bubbles resonate. So they have a very tight, this is scattering strength for a single bubble versus frequency. So if I have a half millimeter bubble, 500 micron bubble, it resonates at exactly 8 kilohertz and nowhere else. It's like a cone. I can pick out a single bubble size with a single frequency. If I look at the other frequencies, they correspond to different bubble sizes. So if I have a little bigger bubble, it resonates like a bigger bell, like a bigger tuning fork at a lower frequency. If I have a smaller bell, smaller tuning fork, it resonates at a higher frequency. So now I can see my 8 kilohertz, maybe I had more bubbles at 8 kilohertz. I don't know. Maybe I had less bubbles. And maybe out on the ends of the band where I had lower scattering, I had fewer bubbles. So that's exactly what we can do. We have a volume scattering strength measurement. We have a model for an individual bubble. So if I have the total scattering, and I know how much scattering per one bubble, I can tell you how many bubbles there are. So now I can make a distribution. I actually can pull out a distribution of bubble sizes from my acoustic data. This is number of bubbles per cubic meter versus bubble radius that I've gotten from my acoustic data, and it shows up here. So I've got 3 different experiments here, 3 different runs, and they have different sort of levels, but they all have the same log normal shape. So this is kind of interesting. And this has been seen, or at least this kind of maximum at about 300 to 500 microns has been seen before. So this is not new, but the shape, pulling out that entire shape is new. What else to say here? Oh, you're going to look at this and you're going to say, oh, Tony, that's like 2 bubbles per cubic meter. Why is it so high? Well, the individual bubble size, the scattering strength of a single bubble is huge. So you can have just a few bubbles in your volume and it's going to be huge. Okay, so what we did was this guy. So if we make an assumption that all the scattering from this, because bubbles are so strong, comes from the seagrass, we can look at the data that comes from the seagrass because we had more time. We didn't have to chop it off where it was in the water. And I can estimate the scattering strengths down here. And so if I do that, let me stop here. I get this distribution. Still log normal, but it's actually got more larger bubbles. So this is got heavier tails than the previous distribution. Same typical shape, except now we're almost 3 orders of magnitude more bubbles. So again, different seagrass from the paper, but you see there are a lot of bubbles sitting on this seagrass. There's a lot in the water column, but a lot sitting on the seagrass. And the ones on the leaves are a little bigger. So we're kind of matching what we see here. But this is the first quantitative measurement of a bubble size distribution using active acoustics of this photosynthetically produced bubbles. Okay, thank you. So we got started a little late, but so I'm going to finish this. So when we were in Italy, there was a thing 3 that showed up. This is only 2 or 3 slides, so it won't take long. So thing 3, I just want anyone to tell me what this is. Anyone have it? Not you.
Is it zooplankton?
It looks like it, doesn't it? Looks like you've got a microscope and it's like a petri dish and you see bacteria moving around. Well, it turns out that's not what it is. So look at the scales. So that's 9 meters by 9 meters, right? So what is that? So what that is, there's a technique that people can do it. Several people do it, but Tim's really good at it. He's at the Applied Physics Lab, University of Washington, called circular SAS. So you do that by beamforming around in a circle. So you're collecting your data and you're forming your final beamformed image by moving around in the circle. So if you use all of the data around that circle, you can take this is a normal SAS image of some really small unexploded ordnance. You can really pull out the detail. The shadows go away and you just get the bright edge scattering. So these are actually bullet-shaped things in a bottle here. So what's going on here? So Tim took 10 circles. The sonar is moving 10 times around over 30 minutes. He's actually forming sub-apertures over a quarter of the circle, just like we did with linear SAS, and we made our movies of the internal waves moving back and forth. He's doing it around a circle. So he ends up with 40 frames over 30 minutes going around an area that's 9 by 9. This is a target, and this is just a cable connected to the target. And these, what are those? Those are animals. Those are bottom-dwelling animals. It looks like they're moving fast, but this is over 30 minutes. So they're really moving about 1 centimeter per minute. So these are crawlies. I don't know what they are. We need to do a better, they didn't collect this data for this purpose, but if we'd had a camera, we could see what was down there. Could they be little crabs or snails? I don't know. But this does let us know that we can actually use this for ecological studies or to look at something like temporal coherence. This is modifying the seafloor. And Tom Blanford has a project looking at temporal coherence of scattering the scattered field from the seafloor. And this could help us. We're good at measuring water column stuff, but we're not good at measuring animal stuff. So this might help us. So Tim's going to visit sometime in the spring, and we're going to talk about maybe collaborating. Just another image. It's the same image, but it's bigger with some pullouts. All right, so that's thing 1, thing 2, and the new thing 3. So I'm done now. Thank you.
Where's those? When I about the boluses at the beginning, it seemed like in the lens, the diagram of the refraction through the lens, it looked like it only talked about the path of the rays. It goes like from the emitter to the seafloor. I was wondering, is that all you need to show because it like does the same path backwards on the Okay, so you don't have to worry about it different.
It's called
because the
reciprocity.
time scale is small.
Reciprocity.
Yeah. Okay.
Look it up.
I know reciprocity is. I was just I guess because you can do that though. I guess just because it's moving so slowly compared to.
That's right. So that's a key point.
Okay.
When doing SAS imaging, the assumption is that everything stays exactly still. If it moves, everything blurs. So if you didn't notice, I should have said, when you looked at the full band image, it's blurrier than the subband images. Because the subband images are as if you took a smaller aperture over a shorter amount of time. So they kind of sharpen up with the boluses.
About, what do you suppose the sort of low frequency sort of shimmer is to the upper right? No, the upper.
No, I'm telling you what I think it is.
Oh, you think it's internally?
I think it's the water column effect. Yeah. Tim said fish, and I don't believe it. But I think it's, sorry, Tim. I think it's a water column effect. So remember, he's forming these over, he's slowly moving around in a circle, and any water column change is gonna maybe cause something.
I believe Tim.
I know you do. I actually should believe Tim before I believe myself. He'll correct me when I see him.
It does look like looking at the surface of the water, it looks like waves.
We take burst images, and there was a fish like in 3 of the images, and it looked just like that, like a ghost. If you average them.
Yeah, they do look like a ghost.
So maybe this is similar to that last question about, we're having to do with the parallax, and you're estimating heights by occupying different positions looking at the same object. So it takes time to get from one place to the next, and that bolus has moved. Can you estimate the speed of the boluses doing that? Or does that just add to the error in your estimate of the heights
That's
because of that time lag?
That is the first question. That's an excellent question. Super observant. The first thing that all of us are like, well, let's just do that. We can track it. Told Jenna, let's see if we can track it. Roy tried it. We've all tried it. These things are a little too amorphous to track forward motion over the integration times, which I think are sort of a little less than a minute. So you can't, they move, they're blurry, but they're kind of blobs moving. And so you can't quite take one look and look sometime later when you were at this time, this position, and see anything clearly has moved. Sometimes we can squint and say, oh, it moved, and then we might say, oh, it moved backwards. So it's just a little too mushy.
So it doesn't really come into play for the uncertainty in the height estimate that you get?
I don't think so.
Yeah.
The uncertainties in the height estimation, there are other things that cause it to be much higher than maybe that. This is kind of a newish thing, and I think we're just getting sort of to where we believe these numbers. When I first saw, 18-meter high things and these volumes of thousands of cubic meters, I went, that can't be right. But they are. If you go to the literature and look at these internal wave boluses, they're that big. And the cool thing about this is we can actually kind of get what's happening laterally. And the papers really just either have a sensor out there.
Don't blind yourself.
You don't like that.
It's going all over.
Look at the reason with remaining eye.
Cool, thanks.
I'll set this down.
So those are like true boluses that have a much more distinctive peak, but you can track internal tides. Would you be able to pick that up, do you think, with this?
You don't see them, you need enough curvature to focus, right?
Yeah.
And so that's only from a bolus. So things like sloshing, the way it manifests is, if you looked at, it doesn't really script the intensity too much, but it messes up the bathymetry somewhat because you've got the wrong sound speed.
Yeah.
So you'll see, I didn't show them, but if in the repeat passes, sometimes you may not see a bolus, but you'll see maybe this water has come through and you'll see that it's kind of depressed. So it's another, if you think you're going to do hydrographic bathymetry with the SAS system, these low angle systems, they're really affected by any sort of inaccuracies in the water column. And we make a lot of assumptions. They don't actually go out and measure sound speed all the time.
How does fare with the double diffusive instability? Would it be possible to capture any of the double diffusive finger instability?
Capture what?
Double diffusive instability.
I doubt it. I don't know. I know what you're talking about. I don't know. Maybe. So all we're seeing here, you have to have sort of an acoustic effect. And I know they've seen these with multi-beams, the staircases, but it's going to have an effect. And one of the images, the one taken off the Oceanographic Explorer, that might be actually something like that. There might be a staircase pattern in the property so that you get a little layer. Because those little ripples on the image, the thermocline was up there at 30 meters and we were down at 90 meters. So there's something else going on, which might be, you can't pick out all of them, but you can, it's having some effect that maybe, that was my guess. Yeah, good comment.
I have a question that might be more for Jenna maybe. For the MATLAB thresholding, what did the three images, like the three colors represent? Was that the progression of the bolus over time?
The two colors, the blue and the red one?
Yes.
Yeah, that's just the, I've identified my blobs, and
Okay.
one's red and one's blue.
It's an anaglyph. If you've done the 3D glasses, have you ever done
Okay.
it? So if you put on the anaglyph glasses, they have one red lens and one green lens.
Let's go 3D.
And you see a shift, and if it pulls it out in a 3D image.
Okay.
But if you just show it the way she did, it just shows you that they're not, the two have shifted because you're looking from different
Right.
directions.
Okay.
It's
It's called what?
anaglyph.
Okay.
And it's really just, it's a nice visual purpose to see like, okay, I've picked them out properly and they've shifted. And then I go along and do my 2D cross correlation to figure out why.
On those plots, the last 3 points at the very end looked like they were off of the curve. Is there like a general effect that could be causing that, or?
The speed plot?
The tail on the distribution.
Yeah, the bubbles.
Oh, yeah, well, where are you? Thank you, I like this.
You want your results to be perfect, don't you?
So I think those are, so those were towards the big bubble sizes, which were the low frequencies. So we have fewer bubbles. My guess is we're being starting to be corrupted by the real seafloor. So we're getting stuff coming in. So, or we're getting down to signal to noise, or so those I don't believe as much.
Yeah, back to the boluses and the magnifying effect. It seems like you're effectively getting like higher resolution within a certain range, within a short area within the say no.
You get worse resolution.
Really? Well, but so part of it's being.
You mean
you're
in the
missing
focus
kind of-
area? You think we get higher resolution?
Well, I was assuming it seemed to that, but no. You're saying no. Okay. Why does it get
so Nick, his thesis, part of it was on trying to understand what the effects on the imaging sonar do to these guys. So what happens is your phases are messed up because your timing's messed up because it's moving in a way. So when you do the beamforming, you assume a sound speed. If you have anything different, if you change the timing, you change the phasing, and so when you try and collapse all the data into a single point beamforming, it doesn't work very well. And so your energy, every pixel has sort of a spread to it. So he found probably 3 to 4 times degradation in the bright spot. So it's actually worse resolution in the bright spot. Because you're using the wrong assumption. Your timing's off right there.
Andy, yeah?
So when you use these SAS sonars for military purposes, would you use multiple runs? Because the sort of regular side scan way to identify whether these things are real or not is often, are they in the same place on passes at different times? Do they do that, or are they just like one time through because they've got such wide coverage?
Yeah, so there, if you're asking me what the military does, I don't know exactly, because I'm not military. But my guess is that they're trying to clear an area or understand an area. So they're gonna mow the lawn to get as much area coverage in a given time. You can, if you do this little trick of multi-looking, it pops out. If you have anything moving on the seafloor, it's because it's something in the water column. So you can see them in a single pass. It just adds a little complication. But Where they do multiple passes is another technique or another reason. So there is a lot of effort going into that. It's for change detection, just like we talked about. You're going to map an area, and then you're going to go back next month or next year. And oh my goodness, somebody put something there, right? Or a storm came and there's now a Fiat sitting out in the water. So they do that. That is a thing. But two different reasons.
Does the multi-look process afford the ability to remove the effect? Like you probably can't get rid of the shadow, but maybe you can actually. I don't know. I'm just thinking out loud.
What I would do, because they're kind of big, is you can normalize the data. So as long as they're bigger than what you're looking for, you might be able to just. So these are uncalibrated, right? So the way we usually treat these images is we kind of normalize them first, right? All of the losses due to propagation and the scattering strength going down, we're going to try and make the whole. You can do a 2D normalizer, but if they're the same size as the object, that's an issue, or near the same size of the bright spot. So if they're big, if they're meters wide and your target is a ½ meter, you might be able to just remove the bright. What you can't get back is signal. So your signal to noise in the shadow is really bad. It's 10 or 15 dB bad. You're actually down close to the noise level. So you can't see very well in there. For change detection, it really screws it up. If you have a target in the bright spot, it's going to be bright. If you have a target, so it really messes stuff up. It's a clutter as well. So these are really not. You want to really not have these. Really, that's the answer to that.
So you don't get a view into the dark spot from one angle that you don't get into another.
Yeah, I mean you do. That's the way to do it, but you lose resolution when you do this multi.
Yeah, let's thank our speaker once again.